Now it's time, it's already time to start our 8th session of the conference. It will be towards marine autonomy. And our first speaker is Charlotte Hauser, professor from bioscience. Her talk will be about robotic applications in coral restoration.
Wonderful. Okay, so that is all this one, yes. So good afternoon, everybody. I hope you had maybe not all of you, but some of you had a great snorkeling trip or diving trip in the beautiful Red Sea. My name is Charlotte Hauser, you heard it already. So I'm actually not at all a robotic expert. I'm neither a marine scientist, but I'm a chemist and I have background. My background is also in molecular biology. And I'm beside that one, I'm here at KAUST as the chair of the bioengineering program because I'm mentioning that one because what I'm presenting to do today here to you, what we are doing in my group on robotics in terms of exploring applications, biomedical applications, marine applications. This is nothing else than doing bioengineering approaches, actually. So that's classical bioengineering. And I would like first to thank Professor Eric Feron, who is here, Professor Shinkyu and Professor Raquel Peixoto, who are actually also collaborators somehow with us, and for this opportunity. And let's see what I can tell you today about robots in the world of 3D printing. As said, it will be, if you will have in the Q&A sessions, technical questions. Fortunately, several of my team members are here. So, and I can refer to them when it gets to the engineering, true engineering part. So let's see. As said, let's start now. You can hear me, I guess. Well, so speaking about biomedical and marine applications and what can robots do there. So here you see a 3D printed, robotic 3D printed coral structures. And in the next, so something very concrete not made from concrete, but some hard material to touch. And here you see something completely different, a 3D bioprinted brain constructs. And so these 3D bioprinted brain constructs, this was done by a team member who's, I don't think she's here today, Shireen Abdulrahman, was copying a mouse brain of this size. So in size and in complexity trying to come as close as possible. And you probably agree that the brain and the coral has not so much to do with each other. And already it's not only in the material properties, there is a difference from these structures I showed you before, but also in the wording. So 3D printing and 3D bioprinting, I hope you know that this is a difference. So it has something to do with 3D and you can do it with robots, but the bioprinting even ends on complexity because bioprinting, you only call that one a bioprinted material when there is any type of living entity included, let's say a cell or tissue or something else. So the other one here, when it says bioprinted brain construct has nerve cells, for example, and endothelial cells included. So what can robots do in 3D fabrication? I have to stay close because this is a microphone. Well, there are challenges, obviously, in traditional 3D printing. If you print, and this is mostly what you do, I mean, there are more and more sophisticated ways actually nowadays in printing. But the classical way of extrusion printing and layer by layer printing has big issues in doing printing highly curved structures. Complex structures. And that would need quite a bit of time to build up this kind of curved geometric constructs. Then you will have a problem if you have material which is not isotropic, but anisotropic. And that makes it also more complex. Quite often, or actually most of the time, in all time, the printer has a designated printing area. So there is a size restriction in the printing in general. Don't quote me for concrete printing, because you probably all know that now in China and some other places in the world, they build even and print houses. So that one is obviously a big printing area, but that is quite dedicated printers and not the general printing we are talking about. There's also an issue with the resolution and accuracy of doing printing, and something which how to polish and finish the surface. Also, the printing times are issues which we have to deal with. So advantages of using robotics for 3D printing is that exactly that one, with that what I mentioned, these curved surfaces or more complex surfaces. So not the classical planar surfaces can be printed much easily because you have a much higher degree of freedom with robots generally. Also, mobility, on-site printing is favored by robotic printing, scalability, flexibility. The resolution and repeatability is also can easier be handled with robotic printing. And last not least, the production time. So before I come and what I come to the corals, let me first look and talk about 3D bioprinting. And you might think about, not because I'm in tissue engineering, this is maybe my main task, what I'm doing or have been done over the many years before I came into coral restoration. But also, you probably know that the coral reefs and the marine scientists will can give more details to this one. This is not only talking about a skeleton, if you do talk about printing. So mostly when we say we want to reconstruct coral reefs, we think about the coral structure. And we heard how difficult it is, how slow it all grows. And then using robots for this one. And that's when you saw, I remember in the very beginning, the structures you can do. This is the skeletal coral structures. But the coral reef has much more. It has actually, I was told it has animal species, plant species, microbial species. It's from everything, something. So it has also tissue. So one of the ideas which nowadays labs are looking into is actually printing not only the hard skeleton structures from the coral reefs, but also looking and printing soft structures. Maybe the polyps, and the polyps are already just not only the polyp by itself, but are in symbiosis with algae, etc. So that's why the tissue engineer or the tissue printing is also a relevant issue for coral or reef reconstruction. I will now focus more actually for true tissue engineering and mentioning something which we have since many years. We're working on this approach that we, with the help here of colleagues, team members, which I could, agreed to support the team from the electrical engineering side, mechanical engineering side, software engineering side, in order to make sure that this system, what I call here the twin print system, which we recently developed, robotic 3D bioprinter, with the aim to fabricate multi-material constructs could be done. So so we call that twin print, and we have published it recently. I forgot to put the citation on it. This is a dual arm. You see here 1, 2 arms. This is small scale, simple lab scale approaches, but in the lab, it's where it starts. Later, it can be upgraded to more fancy systems, obviously. So you have the hardware of 2 robotic arms, 2 sets of microfluidic pumps, then very important, the nozzles, 2 quadrupole coaxial nozzles. And then, of course, the software also is important. So integrated software, they were developed in-house. So we have here the experts around. And if there are questions, fortunately, they will probably then help me later for the questions. These software should also then control and synchronize and make this multi-material 3D printing more easy. So, so how does twin print work? Well, I will show you soon a little video, just short video, how you see how 2 arms get synchronized into a small little construct. So why why multi-material actually? So the whole thing is said multi-material, multicellular printing, and that is not only with us as a human being where we are very complex in all terms, even simple skin is not that simple. But this is also, as I just mentioned, with even the coral reefs, the same. So there are symbiotic systems of different species integrated. So multicellular systems are also there. Tissue is also there. Multi-material is also there. So that one, what we do for classical biomedical applications could apply as well for coral reef restorations. And then, of course, all needs to be integrated. So why we use multi-material, as said, the goal is here to overcome the complexity of all these different components which are in there, not a set of different cells, extracellular matrix, biochemicals, and et cetera, et cetera. So here is now the little video where you can see these 2 arms. Let me see how I can get it. Got it. Yeah, started. 1 arm this is in a greenish, in the transparent way. Maybe I make it once more again. It was a bit quick. Oops. So one comes the greenish material, the transparent greenish, transparent greenish, transparent, and then, oops, it's at the end. So you see, it's a little bit hard. The focus is not so much on the structure, but you see even that one has a defined structure. So a printer not only is the hardware, the software, you need obviously, every printer has an ink, so it has a material what you need to print. And as we know, when we buy laser printer, whatever, mostly companies sell their own material, their own printing material, so it goes along with the printer. There are very few printing systems available which can use actually different types of inks. And for a bioprinter, you need a bio ink. That was actually the beginning of the idea doing bioprinting, that many years ago, I started with a very interesting system. And I told you I'm a chemist from background. So, and later went into molecular biology. So I came, that's around more than 10 years ago, almost 12 or 13 years ago, into encountering two small, interesting peptide compounds. And a peptide is nothing else than a little part of a protein, and we are made out of proteins, defined by the number of amino acids. So this one we call ultra short because indeed the size of these compounds, they have just a handful of amino acids, 3 to 6. Generally here at KAUST, we work with 4 amino acids. So that is really a handful numbers of amino acids. And these compounds, this is just a kind of way of how we do it, have what's called an amphiphilic motif. So a hydrophobic, a hydrotail, and a hydrophilic head. And so they gets quickly in a polarization state. That actually drives something, what I mentioned before, or what you see here, a process which is called self-assembly. Self-assembly is just something what you see. We know mostly chemistry or biochemical synthesis of how we are getting, how we are made of, but we are made also of self-assembling systems, like the lipid membrane, the DNA, how it assembles. So these compounds, what it just simply means is that that molecule knows how to interact with each other, how the molecules know how to interact with each other. They recognize each other, they signal with each other, and they need nothing else than just maybe just water to make these interesting structures as here of these, what you can see here, fibers. So these fibers, and these are, we are speaking here in micrometer scale, nanometer in the single one, they string together. They are part of here, this system here of pores, of the membranes of these pores. And that is nothing else what you see here in that so-called soft solid hydrogel, which is made from that synthesized compound dissolved in water. So magically they get together, form these kind of strings, etc. And so this kind of behavior was, when I looked more deeper into it, I was getting really fascinated, not only because that material has unique properties in terms of being temperature resistant. You can heat it up to 300, which you normally can't do with protein. They coagulate. This material can really quickly, and generally proteins coagulate, so they don't survive big heat. This material does have a long shelf life. They have also, by being obviously biocompatible and interestingly also non-immunogenic, they have very interesting, unique optical and even conductive properties. I'm not going into that one now, but saying that we can synthesize, that means also we can tune it and modify it. So we can add on the so-called basic construct, the basic molecule, the 4 amino acids. We can hang on a lot of things which we want. That's saying biofunctionalities, etc. And we can make it also that this material, these gels are actually getting more or less viscoelastic, more or less adhesives, etc., etc., degradable, etc. The interesting part, and that's why I'm talking, so I wanted just to make you aware of this, of that stuff. No, goes to the back. We didn't want to go to the back. I have issues with the mouse. Yeah. So here is a material, this solution extruded with a syringe, and blue is just a food color on a fluid which has some buffers in it. And you can say we created so-called, it solidifies instantaneously in so-called, we call that noodle system. That was the birth of saying that material could be used as a printing ink, as a peptide bio-ink. And we don't own, by the way, have just one compound, we have many. It's a whole class of what we have, and we can modify it with a lot. With chemistry, you can do a lot. So due to that low viscosity properties and that they instantaneously gel, as I could show you here, with very low concentration of material, that was when we triggered and we said, oh, let's see, can we do something with printing? And as for the biocompatibility, for the tissue engineering purposes, that triggered it also when we knew, you learned more about the properties, because believe it or not, and I don't want to go into here now really in biochemistry or in biology more, but these fibers have very strong assemblance to collagen, and collagen is a structural fiber protein in our body, which holds things together. So anyway, these kind of all this knowledge, sorry, going in the front, let us build up a very nice peptide platform technology for a lot of different applications. And you see here imaging, injectable therapies, wound healing, etc., etc. Let's focus just here on the peptide bio-ink. So we can do different patterns. You see again that little thing where we all started with, this robotic small robot arm. And we could create a human nose kind of structure, which also has this curvature, which just, it's easier be done by a robotic printing. And then here's constructs which are prone and can be used. And we have a project here on the trachea, for example. We have many other ones, the brain I mentioned in the beginning. So I don't want to go further on that one, but now let's get back to the marine life. So it was not us, but others. We actually call it Daniel Wang Prasert from San Diego, University of San Diego. I think he all the scripts, who did for the first time a 3D printed microalgae in a bioprinted one, so not printed one. So that's getting back, combining into the thoughts. Also, not only tissue, not only skin or whatever can be bioprinted, but also marine tissue. So we aim actually to use this twin print system for coral fabrication, coral reef fabrication. How we mean that one? So that would mean that we can fabricate the coral skeleton with one arm, with an ink, which we also create, and I come to that one soon. For so the skeletal structure, but then apply also something like adhesives or other protective substances, maybe hindering macroalgae to grow on, and adhesives to glue microfragments on, and then to do even probably bioprinting of soft body coral microorganisms on top of it. So that could be done independently from all these different arms. So now, having that small size of arms in our lab, we also looked for bigger approaches. And we got into these nice industrial arms. We could purchase this one. It cannot be in the lab standing because it's kind of huge and it has this size here, almost man size, and the arm, and it goes up and it goes around. It would be too dangerous to have it going around. So this one, we wanted to see, can we use that also for a so-called twin, in the future at least, twin print system, or just explore it, because this is just a robotic arm, not a printing system. Can we make these arms to a printer? And actually we succeeded, and I see now here Shinkyu, for the Shinkyu Park, yes, hi, who helped here a lot to get something done, which to really then print a coral brain structure. So we started with to produce a hybrid fabrication process and do coral structures and molds in order to see with a specific ink, I mentioned that soon, which can be used to do coral fragments, and then deploy hopefully our adhesives at one time with these two arms, and then also exploiting biopolymers for coral growth. So the system has a high DOF motion and large workspace, as you can see here, faster way than the small ones for sure to do constructs, to produce constructs, and is full automated. And that is a mini brain which we could then actually print. And I think I have a little, no, not yet. So again, the advantage of non-planar additive manufacturing, the classical traditional layer by layer printing is really overcome with this robotic printing by multi-directional fabrication and a reduced building time, and much better, particularly with these huge arms, large-scale fabrication process. So we did in the beginning, this is just PLA, polymer constructs, which we printed then with this printing arm. And this was not a printer, as said, a robotic arm. And then we were able to get that one here. Oops. Sorry. Yes. How did I get it? No. I would like to. Yeah. So now you see this one. This is, of course, not in real time. So this is the process which we did in collaboration with Professor Shinkyu Park and his team members. And you see soon how a little brain coral fragment is constructed. And that is not any longer a polymer. So this is something where we also created our own material, our own ink, coral ink, which we call CCP, calcium carbonate photo-initiated, sorry, calcium carbonate photo-initiated ink. Now you see it here at the end. So this is the little brain. So in order to see what we have, there is a lot of factors which implement or which are impeding coral restoration: cost, footprint, carbon footprint, manpower, scalability, effectiveness. And I will just quickly run through and not go too much into detail, but we also had published recently with other colleagues here at KAUST, Professor Duarte and Manuel Aranda, Sebastian, I saw here as well, the audience, a 3D Cora print principle, where we used specific material and a way of doing these structures, which I will show you soon. So, but just has 3 phases: replication phase, fabrication phase, propagation phase, and we're running out of time soon. This is a reverse engineering process where you would go through this process: harvesting, scanning, modeling, CAD modeling, editing the model, preparing for the printing. Now we have an LCD wet polymerization process, and then the so-called in-house CCP, which is nothing else than something very nice, which comes in particular ink, which can then do these structures here, which or others, and I will show you these ones like here, these kind of coming close to branch structures, the brain structures again, where you can deploy or adhere with specific other material, the corals, and you see how nice they adapt to it, how nice they grow. And this one is actually still the very beginning. It was a polymeric structure, which we still have. Now it's over 3 years here where we have it in the tanks, and it really increased largely in size and in matter, in structure. But the aim is using the CCP ink, and that is what we did, because it is copying the surface and the material of a coral reef, the skeletons. Sorry, this goes back. And you see that we are trying really to come as much as close to the real. This is the native coral. This is the printed coral. Here you see it, how it gets deployed. And that is now after deployment here, how it's grown now in the sea, in the field. And I guess you had also Professor Raquel Peixoto and her coral vigil, where she really did a lot of efforts to establish this here at KAUST. And we have a place also in the coral village with our deployed structures. And you see how over time they look and they grow. And with that one, actually, I would like to stop here because the time is over, and just would thank all the team members here, where some of you are here around, and then the collaborators here. We have Eric and Shinkyu around. There are Sebastian as well. There are others here who have contributed, and particularly thanks to the group here and their contribution. Thank you so much for your attention.